My name is Bruce McEwen. I'm a professor here, and I'm here to welcome you on behalf of the Rockefeller University and also the Science Outreach Program to what is the 50th year of the holiday lecture, which used to be called the Christmas lecture. This lectureship was established in 1959 by Alfred Mirsky, who was a professor here, and was modeled after the Faraday lectures for young people that were started in London in 1826 by Michael Faraday, who was one of the greatest experimenters in the history of science. Some of you may have seen the book called The Chemical History of the Candle, which is one of those lectures. The holiday lectures become the centerpiece of our science outreach program that was established in 1992 for high school students and teachers who work in labs and form a partnership with the lab and with researchers that carries over into the school year. And I'm sure some of you have perhaps been part of that program. There's a brochure that you can pick up outside on the outreach program. Now, in 1959, the first holiday lecture was given by René Dubos, a famous microbiologist and also environmentalist, and was called The Unseen World. Now, our bodies possess an immune system that protects us against the unseen world of pathogens, and it's fitting that this year's lecture will be given by an immunologist, Nina Papavasiliou, who studies how the immune system does its job by rearranging sections of its DNA to form specific antibodies. Now, Nina was born in Thessaloniki, Greece, and won a scholarship to Oberlin College in Ohio, which happens also to be my alma mater, so I'm particularly pleased to introduce Nina. Nina majored at Oberlin in biology with a minor in German literature, graduated in 1992, was admitted to our Rockefeller's Ph.D. program, and got her degree, Ph.D. in 1998 in molecular immunology. After postdoctoral work at Yale, she returned to Rockefeller in 2001 as an assistant professor and lab head and was appointed associate professor in 2007. She has been a Keck Fellow, a Searle Scholar, and won the Alexandrian and Alexander L. Sinsheimer Fund Scholar Award in 2005. So without further ado, it's a great pleasure to introduce Nina.
Okay. Hello, everybody. It's great to see that many of you here today. It's supposed to snow later on. What we're going to talk about today, as Dr. McEwen alluded to, is about mutation. We're going to talk about mutation and about how it is used by our immune system to protect us against foreign invaders. And in the second part after the break, we'll talk about how mutation is also used by pathogens to evade the immune response. So we're going to start with a few definitions. First, some philosophical definitions about what science is and isn't. And second, we'll define mutation. And before I go any further, I want to make this quite as interactive as possible. And so what I'm going to do is I'm going to be asking you questions during the talk. And I actually have some presents to throw out. This is a white blood cell. Whoever answers questions during the first part of the talk gets a white blood cell. Hopefully I'll be accurate in throwing them out. And whoever answers during the second part of the talk will get this parasite, which will become clear what it is later on. Okay. So at the same time, I would like you guys to ask questions as well, either at the end of the talk or even during the talk. If you have a particularly good question you'd like to ask, just raise your hand, and I will call on you, and I will also repeat the question to make sure that people watching the webcast, as well as the rest of the audience, can actually hear what you have to say, and also to make sure that I understood it correctly. All right, so we're going to start off with my philosophical, at least, definition of what science is and isn't. So normally, when you read science or you learn science from the books, science is presented as a body of knowledge. That is, facts to be memorized, equations to be solved, concepts to be understood, and discoveries to be applauded. And that is partly what science is, but the working definition of science is not this. This is what you learn in school. Science, and you have to be, of course, comfortable with learning, but at the same time, science really is about what we don't know. And there's an article of which I have some copies here, published a year ago, which is really, really good, and I recommend it to anybody. Again, if you'd like to pick up a copy, I have some here. Recommend it to everybody who is interested in getting into science. Science is about what we don't know, so you must be able to get comfortable with being ignorant, and as the article says, being stupid. So the article is titled "The Importance of Stupidity in Scientific Research," and you're welcome to pick up a copy. So science is not only about what we know, but mainly about what we don't know. Science is also not a quest for absolute truth. Absolute truths don't really exist very much in science. Much of what we thought we knew in the past, and I hope that will become clear during the talk, has turned out to be incomplete or plain wrong, and this will happen again and again. Science is really only about observing nature, making models based on those observations, and testing the predictions that these models make. And if these models are good and useful, they make good predictions, and these predictions can be proven or disproven, and each one of those is equally valid to science. Science, unlike what we usually read in books or even in scientific papers in the literature, is not a logical accumulation of evidence. That is how it's presented because it's easy to understand, but science actually benefits from non-linear thinking, and as you do science in the lab, much of the best science you'll find requires leaps of imagination and speculation. And in fact, I write here daring speculation, but as you'll see during the talk, especially the first part of the talk, the leaps are not that huge. It just takes a few people thinking outside the box to actually reorient science. And finally, and this is at least my belief, science does not necessarily have to be of immediate or direct benefit to humankind. It doesn't have to be what is commonly thought of today as translational. Science is really about learning. And that means that nothing is too fanciful or exotic, even if it doesn't seem entirely relevant. And I have a few copies of a paper published just a couple of months ago, and called "How to Learn New and Interesting Things Based on Exotic Biological Species." That is, science as we do it today is mainly based on knowledge from 5 or 7 model systems that we have been working on in the past 50 or 60 years. But model systems exist because people made them into model systems. And there's a lot of biology that could be quite relevant to disease out there that exists outside these 5 or 6 or 7 model systems. So learn to think outside the box, learn to love science for the learning of it and not necessarily for wanting to directly translate it into a benefit to human disease or humankind in general. All right, so now we're going to go into the main part of the talk. We'll first define mutation, and this is the first question I'd like to ask. These are 4 possible definitions of what a mutation is, and I'd like somebody to tell me whether a mutation is a base change in DNA, a permanent change in the sequence of a gene, the force that drives evolution, or an independent record label from Alabama. And this will be yes.
Let me guess, all of the above?
All of the above. That was an easy one. There we go. Okay. That was easy. It gets a little harder. Congratulations. That's a good guess. So when is mutation useful? Mutation is very useful at the level of a population because it helps produce genetic variation. That is, imagine you have a pond, a saltwater pond, in which algae live, and these algae live quite happily. There's a very large population of millions or billions of them, and at some point something happens, and instead of salty, the pond turns very, very acidic. That acidity will kill off the vast majority of individuals in the population. But if there is a certain amount of mutation-produced genetic variation in the population, the individuals that are mutants will actually, or may be, able to survive the new environment. And so mutation in a population helps produce a type of the genetic variation that will allow the population to adapt to new circumstances. So mutation is actually quite useful for survival. However, At the level of the individual, mutation is random, and that means it is almost always detrimental. However, there are some situations, which we'll talk about later on, where mutation is clearly, at the level of the individual, also beneficial, and that is in addition to Marvel comic type endowing humans with superpowers. All right, so work on mutation starts really with the paper published in 1927 by a fellow by the name of Hermann Muller. It's a really amazing paper, and I have a couple of copies if anybody's interested. It's called Artificial Transmutation of the Gene. And what Muller did was to X-ray fly oocytes and to then make those flies and generate mutant flies of many, many types. And what he noticed, as these flies are coming out with more than the regular complement of wings, or 4 eyes, or 3 or 4 or 5 antennas, what he noticed, or many more legs than normal, what he noticed was that these mutants were actually observed in nature as well, or at least some of them were. And therefore, that mutation was induced, these X-rays were actually accelerating a normal, spontaneous, natural phenomenon. So at this point, I would like to ask the second question, which is, can somebody tell me what a gene is? Can somebody give me the definition of a gene? Okay, over there.
It's a group of DNA clustered together so that at the bottom part of the chromosome, all the DNA.
Okay, this is a valid definition for 2009. However, in 1927, it was not clear for another 20 years, really, that genes were made of DNA. So before you knew that the gene was a piece of DNA, what did Hermann Muller think of as a gene? Okay, way back. Was that a characteristic? That's right. Oh, now you have to. Can't throw that far. Let's see. Oh, I'm sorry about that. Wrong person. You have to come get it. That's exactly right. So a gene is a trait. A gene for Muller is a trait. It's a characteristic. It's a phenotype. And this is important because oftentimes in science we don't think that the people who have made an observation much earlier on were not used to thinking the way we are now because they just didn't know the same types of things. All right, so there are a couple more observations that Muller makes that are quite astounding for this being 1927. One of those observations is that the mutants he was looking at, if he looked at the level of chromosomes, had rearranged chromosomes. The reason he could see this is because flies in certain tissues like the salivary glands have these huge chromosomes that are produced by enteroduplication. And these chromosomes have a banding pattern. And the banding pattern is actually like a fingerprint. So that if this black band here were actually removed to another part or duplicated another part of the chromosome, you would actually be able to see that by a microscope. And so what he observed is that if you looked at the chromosomes that these mutant flies produced, there was actually a shuffling of these gene segments. And this shuffling of gene segments corresponded then to a shuffling of traits. And so about 80 to 90 years, 80 years before the human genome was sequenced and assembled, Muller makes the observation that this type of recombination can be used to create chromosome genomic maps, which is actually an astounding observation before people knew what a gene was. And then Muller finishes the paper by saying that in conclusion, attention must be drawn to the opportunity of creating a series of artificial races for use in the study of genetic and phylogenetic phenomena. And he finishes off by saying that the time is not ripe to discuss such possibilities with reference to the human species, but comic book writers didn't listen to that. And so as a result, Marvel came up with a bunch of comics, and I'm sure you've seen many more than these. X-Men comes to mind, where this bunch of fellows is hit by cosmic rays, which make this rack-tack-tack-tack sound, and they're not properly shielded, and so they start becoming mutants in real time. So this guy here looks at his siblings and says, "You've turned into monsters, both of you. It is those terrible cosmic rays." And then he turns into a fireman as well. All right. Now, we're moving on to about 20 years later, 15 years later. And as Muller had already shown, mutations could be induced by X-rays, but X-rays were simply accelerating the type of event that was normally spontaneously happening in nature. And so these two fellows, Salvador Luria and Max Delbruck, who are also incidentally credited with introducing quantitation in biology, in other words, introducing a little bit of math in biology, which up to that point was an observational science. These two fellows decided to ask the following question. What they knew was that if you had a population of bacteria, which you plated on a plate that contained phage, which is a virus that can kill bacteria, the vast majority of bacteria died. But there are also always a handful that survived. And so they wanted to know whether the bacteria that survived phage, the bacteria that became resistant to phage, were actually pre-existent in the population, or whether the fact that they touched phage led to the induction of resistance in a number of them. And they devised the following experiment. The experiment is predicated on starting with a very, very small number of bacteria, ideally a single bacterium, and to grow from that single bacterium a large population of a few billion, and then to plate those billions of bacteria on agarose plates containing phage. So what they knew, as I mentioned a second ago, is that the vast majority of these bacteria will hit the plate, get infected, and die. But a handful will be resistant. So the question is, were mutations, again, induced by phage, or were they pre-existent and selected on the plate containing phage? And so here's the experiment. These are two different experiments, sets of experiments, experimental outcomes rather. This would be the outcome if mutation were induced, and this would be the outcome if mutation were spontaneous. And let me walk you through these. In every single case, you start with a single bacterium, grow to a population of, well, 8 are shown here, but let's say 8 billion. And then you put that on this little agarose plate. If mutation were induced by the fact that the bacteria are plated now on phage, then you would have about the same possibility of getting a mutant bacterium every time you start with a single one and grow up a population. And so basically in this particular situation, these two guys somehow ended up mutating, and you have two colonies on the plate, or these guys, or these guys, or these guys. Now if mutation were spontaneous, the outcome would be totally different. The outcome would be that sometimes you start with a single bacterium, and as you grow it into a large population, because mutation is random, you actually don't have any mutants that have acquired resistance to phage. And so in this particular experiment, growing from a single cell onward and plating on the plate, absolutely nothing grows. In this situation, pretty close to the time you plated, maybe one of the bacteria actually acquired a mutation that made it resistant, and so on this plate, there's going to be a single bacterium that grows. If this mutation was acquired a little bit earlier on in the population doubling line up here, then the bacterium that mutated here will have had the time to make two bacteria, and so those two are going to sit on the plate and make colonies. This is a situation where half a plate would be full of mutants if the mutation occurred very early on during the set of divisions. And so what Luria and Delbruck found out was that the situation was this. In other words, that mutations arose spontaneously during cell divisions prior to plating, and therefore each plate had a highly variable number of mutations, or mutants on them. And so here we are now in the 1950s, and the state of mutation research, knowledge about mutation, was as follows. By now we know that genes are made of DNA, and so mutation is in fact an alteration of the sequence of DNA of a gene. Mutation is inducible, accelerated by X-rays and other means, or mutation can also be spontaneous. And really mutation is spontaneous and it's accelerated by various means of induction. Mutation allows for the adaptation of a population to changes in the environment and is therefore the driving force of evolution. Now in the 1950s, and the experiments that were done with regard to mutation were experiments in single-cell, rapidly dividing populations, such as bacteria or yeasts or algae, or in germ cells, the oocytes of multicellular organisms like the fruit fly. And so all these statements are actually correct except for the last one. And it took a foray into immunology, which I'll go into in a second, for people to realize that mutation is actually something that can take place in many, many different cell types, in multicellular organisms, in somatic cells, so not germ cells, and those kinds of mutations may actually have a beneficial effect for the organism as a whole. And so really what brought what made this statement, what made it clear that the statement was not accurate was what was called the antibody problem. So here is the antibody problem. People knew, chiefly from work done in the beginning of the 20th century, that white blood cells make antibodies. People also knew that antibodies can react with any pathogen or synthetic chemical or bit of protein that one would inject into an animal. All of these are called antigen. And antibodies would attach to antigens and target those for clearance. And the question really was, if an antibody in the body can actually have the ability to interact with what ought to be an uncountable number of antigens that human body would interact with during one's lifetime, then how is it that how is it that these antibodies can react specifically with a huge, huge number of different antigens? And so this became a binding problem of antibody to antigen and gave rise to two competing theories that we'll just mention now and we'll talk about extensively a little later. The first theory, which was supported by, I should say, the vast majority of the scientific the group of scientists at the time, active scientists at the time, was that antibodies actually mold around the specific antigen upon binding. So you see an antigen, the antibody comes, and it actually rearranges to bind the specific antigen. There was precedent for such a thing. This was a heyday of Jacob and Monod, who had already published in enzymology experiments that in many cases, enzymes were actually binding their substrate in a similar fashion. A competing theory was mainly pushed forward by Joshua Lederberg, who has had a long association with Rockefeller and just died a couple of years ago. He was a geneticist. And what he proposed, which is one of those first little leaps of imagination, which in retrospect are not that huge, but at the time were huge. What he proposed was that antibodies and the cells that carry them are just like bacteria. In the same way that I told you that bacteria pre-exist with the ability to be resistant to phage, the same way he posited that antibodies pre-exist that have specificities toward all possible antigens. And so before I explain these two theories any further, I want to give you a very short introduction into immunology. So, the immune system. The main role of the immune system is to recognize and defend against microbial infection. And so what I'm going to show you in the left is a slide of, let's just say, a macrophage going after a bacterium. And on the right, I'll show you a representation of the same thing, which will allow you to see something that you can't in the real-time slide on the left, movie on the left. All right, so here is a real-time video of a macrophage actively going after a bacterium. Whoops, I'm sorry about that. There we go, little macrophage, go back. Okay, here's a macrophage going after the bug, and here is the representation of this. So macrophages swimming in a population of bacteria are going to start grabbing, putting out these protrusions, are going to start grabbing bacteria and eating them. This guy's still going after him, still going, and whoop, now he's eating the bacterium. Okay, so now this movie ends, but at the point the bacterium is eaten, it's not that the cell becomes quiescent, it actually starts spitting out this huge number of molecules called cytokines and chemokines, whose main purpose is to attract a large number of cells of the immune system to the site of infection and eventually clear the infection. So the slide you just, the little movie you just saw has actually to do with the innate immune system. This is the most ancient immune system. It is fast. It recognizes any invader in a very fast manner. It is, however, non-specific. So really, any bacterium of any kind, or parasite, or yeast that goes in through an epithelial layer, for example, will be engulfed and eaten by a macrophage, or by a number of cell types, of which very many exist, that will do exactly this. They will eat up the parasite, the pathogen, and then, or the bacterium, and then will attract the adaptive immune system to the site of infection. So the adaptive immune system is probably less ancient. It consists of two main cell types. These are called T cells and B cells. And it is very, very slow. It is, however, very specific. The reason it's slow is because these cells, after getting these signals from macrophages or dendritic cells or the very many other cell types of the innate immune system, these cells will need to mature after this signal. And T cells mature into cytotoxic T cells, which will go on and kill actually infected cells by recognizing the fact that they're infected. And they will also will differentiate into what is called a T helper cell, which will provide help to a B cell, which will now begin producing specific antibodies to the original invader. And so this lag in terms of a need for development makes the adaptive immune system extremely slow, but the reaction is always very, very specific, and also, as I'll mention in a second, has a memory component. So here's another movie that explains what happens after you've been infected once and your immune system actually recognizes that fact. All right, so here is a bloodstream, and all sorts of cells are going through. And at some point, you'll see a few bacteria that are going to sit. Well, there we go. We're zooming in one of them. This guy is going to sit on the endothelial cell layer of the vein, and is going to try to essentially breach this barrier and go into the tissue. And there are some more of these guys going on, attaching to receptors on the epithelium. But here you go. Here are specific antibodies, because the body remembered that these cells, these microbes had infected it before. These antibodies will both block the microbe from entering and will make it tastier for the innate immune system to pick it up. And I say tastier because this is actually called opsonization, and that apparently is Greek for becoming tasty. All right, so there is not only an interplay between the innate immune system, as in it recruits the adaptive immune system, which then is not only allowing for a specific response, but also memory response. But as you saw, there is also a crosstalk going the other way. These antibodies now can attach to a particular pathogen and allow it to be better cleared by macrophage. And so I'd like to stop here and ask you guys, if you were to design the system and you wanted to use mutation to improve one of these two branches of the immune system, which part would you use mutation in? Yes?
Adaptive immunity.
The adaptive immune system. All right. Why do you think that's the case? Why the adaptive immune system?
Because then you could remember it, and then it'll be an advantage to you most of the time.
Okay, yeah, so it's mutation because, as you'll see, it allows for specificity. If you mutate an entire population of cells, it really is like mutating an entire population of bacteria. And then you can pick different specificities based on what you got infected with. All right, so the main role of the immune system is to recognize and defend against microbial infection. There are two arms to the immune response, the innate and the adaptive arm. And there are two main types of cells in the adaptive response, B cells and T cells. Now, B cells recognize extracellular pathogens directly. So any bit, whole pathogen or bit or piece of microbe that is swimming in the bloodstream or in other tissues, a B cell can recognize directly by using receptors, and these receptors are called B cell receptors when they're stuck on the surface of the cell, or they're also known as antibodies when they become soluble. T cells, in contrast, recognize intracellular pathogens indirectly. That is, they recognize the fact that a cell has been infected. And they do this by using a dual receptor system. So T cells, as they swim around the body, can touch any cell and determine whether this cell is a self cell or a foreign cell. And so they have a certain set of receptors that recognize the fact that a particular B cell here is actually a self cell. And that avoids killing by T cells of cells of the human body. At the same time, T cells have to have the ability to use receptor sets to recognize an infected human cell, an infected body cell. So there's a two-receptor system. One recognizes the fact that this B cell here, for example, is infected, and another recognizes the fact that this B cell is a cell of the body, but an infected part of the body, so now please kill it. So now, if you were to pick whether a B cell or a T cell will benefit from mutation in generating more specific receptors, which one would you pick and why? B cells, why?
They're the ones that recognize, they're the ones that are more specific. B cells are not specific.
It is B cells, but that's only half the answer. So why would you pick a B cell as, yes? Not quite. Over there.
Because it fights the pathogens more directly and space the actual cell.
It's not this. You guys just forget about what the B cell does. Just think about the fact that it recognizes with one receptor versus two. Anybody else? Way down there.
Well, because it avoids killing the good cell.
No, don't. Just forget about what it really does. Just if you had a cell with a single receptor and a cell that required a dual receptor recognition system, which one did you pick to benefit from mutation? Way down there.
Diversity of antibodies, so then to attack different pathogens or infect
Well, a T cell is also quite diverse, but that's still not the answer. Forget about what these guys do. Two cells, one needs one receptor, the other needs two.
The antibodies are changing, could be changing, so maybe the B cells have to change with them, like to be able to adapt to the new antibody.
That is close, but not quite. Okay.
Because what B cells have is antibody, so if there's mutations, so the cells can change with different types of pathogens, so you can help it adapt better.
But why would a T cell not be able to change? Because T cells do change as well. Yeah, the question is, if you have two cell types, forget about them being B cells or T cells, and one cell type recognizes something by using one receptor, one receptor, and another cell type recognizes something by using two receptors at the same time, which set of cells would mutation be a direct benefit to?
If you only need one receptor, then it's just a
Right, so that's really the answer. That if you have one receptor, when you mutate it, there's nothing else you really need. If you have two sets of receptors that are codependent in a way, then you mutate one and you screw up recognition of the other as well. So that gives you a B cell. There we go. Okay, sorry, this was a little complicated. All right, congratulations. Okay, so back to the antibody problem. Now that we know a little bit about the innate and the adaptive immune system, about B cells and T cells, let's go back to the 1950s to see what this antibody problem really is. So what was known about the antibodies in the 50s was defined about 50 years prior to that by Paul Ehrlich. He's a fantastic scientist who first found that white blood cells make antibodies, also described how antibodies can react with any pathogen, protein, or synthetic chemical. Therefore, they could really react with anything specifically. And he also found that antibodies attached to these pathogens, proteins, or synthetic chemicals and target them for clearance and destruction. And so that was what was known. And there are two competing theories to explain the fact that antibodies bound a huge number of antigens extremely specifically. One theory said, and again it was a prevailing theory of the day, pushed by people who were Nobel Prize winners at the time, mainly enzymologists and protein chemists, that antibodies mold around the specific antigen upon binding. And this made a lot of sense, except it did not explain everything. It actually didn't explain a main component of the immune response, which is memory. So why if you were an antibody molding around a particular antigen, how does memory come about? You would have the same chance to mold again around a particular antigen the next time around, and so there would be no memory response. So this did not take care of the memory response. There's another theory that was pushed forth by Joshua Lederberg, who was a bacterial geneticist and also Nobel Prize winner at the time. And the theory was that it's simple. Antibodies and the cells that carry them must pre-exist, like bacteria pre-exist, that have specificities toward all possible antigens. And this seemed quite reasonable to a geneticist that was used to studying bacterial genetics, but seemed quite a ridiculous statement to enzymologists, biochemists, as well as actually immunologists, because for this to be true, certain other things needed to be true, and those were not known at the time. However, Lederberg wrote a very influential paper called Genes and Antibodies, which was published in Science in 1959. And what he wrote was, do antigens bear instructions? Do they mold around the antibody? Or do they simply select, like bacteria would, cell lines that arise by mutation? And of course, what he was really pushing is that antigens, so pathogens, proteins, synthetic chemicals would select out of a population of B cells and antibodies, cells that arise by mutation. And I think it's fair to say that the thought process that shown through this paper in 1959 has really, almost every single bit in that paper has been validated and is actually what we know to be the case in immunology today. So there are 4 corollaries that were put forth in this paper that needed to be true for the statement to be true. And I'm going to go through each one of them at a time because every single one of them ended up being correct. So first corollary is that B cells are clonal, so that one cell will make one antibody. Second is that upon interaction with antigen, the cell that had a specific antibody multiplies. That is because you would need a population of cells upon which mutation could be induced. And that again harks back to the fact that a bacteria, if you mutate a single bacterium, you'll likely kill it. But if you mutate a population, you can be selected for, allow for selection for a new trait to occur. B cells then would mutate in some fashion their antibody genes specifically, that is, mutate the particular bits of DNA that encode for the antibodies while leaving the rest of the genome intact. And that was a pretty radical thing to propose, but ends up being true. And finally, antigens will select from this pool of mutant B cells and mutant antibodies the ones that bind best. So we're going to go through each one of these at a time. So B cells are clonal. This was actually proven by Macfarlane Burnett, for which he got the Nobel Prize. And this was very important because biochemists and enzymologists were thinking of the antibody as a protein. And you cannot really apply selection to a protein. But if you make the tiny, tiny step to a different direction and say, we're not going to select, we can only apply selection to genetic systems. So if each protein, each antibody is carried by a cell uniquely, then you can apply selection pressure on that cell to pull it out of the population. So selection is applied on the cell, not the antibody, and that tiny, tiny, leap of imagination ended up changing the way immunologists thought. So here is a population of B cells with receptors on the surface, and here is an antigen X that will specifically bind and therefore pick out of the population this B cell. The second corollary is that upon interaction with antigen, the cell that specifically binds antigen multiplies. And this was actually known for a while. This was first observed by Paul Ehrlich, who is shown here, for part of the reason for which he got the Nobel Prize in 1908. So what Ehrlich observed is that the cell that binds the antigen actually multiplies into a very large population of cells, actually. And then the third corollary was that on that large population of identical cells now, mutation will be induced in these cells, and mutation will be induced specifically to the antibody genes. And so here is that B cell now that bound the antigen, and here are the antibodies on its surface. And there are two, before anything was known about how this happens, there are two competing theories. One theory was the somatic theory of mutation, which posited that this was really a bacterial system in the sense that there was point mutations on the antibody genes and single amino acid substitutions in antibody genes so that you would actually physically mutate the genome and give rise to a group of antibodies, a group of B cells that are related to one another, but they're mutants of one another. The second theory was called the germline theory and posited that genes or gene segments actually exist to encode each antibody. Because if you think about it, you need to have some substrate on which mutation to occur. And what these guys are saying is that each cell came up with a single antibody. As you came out of the bone marrow, all the cells were pretty much identical, and then upon touching antigen, they underwent a program of mutation. And this was known not to be true. And so one of these theories had to be true then. So here's now what the antibody looks like at the protein level. It is made up of 2 light chains and 2 heavy chains. And it turns out this fellow by the name of Susumu Tonegawa got the Nobel Prize for, I'll describe in a second, as being how antibodies are actually made. So what Tonegawa showed was that this antibody that was made up of different chains, if you looked at it at the molecular level, the DNA level, these chains were not contiguous. In other words, the protein that is the antibody is not encoded by contiguous fragments in the genome. But in fact, it was encoded by gene segments which are recombining in somatic tissues, so in B cells. So this is actually also a radical idea. Basically what happens is you have a large number of what is called variable gene segments, diversity gene segments, and joining gene segments, and then you have a constant region of the antibody. And what happens stepwise during development is this gene segment right here, this D, will recombine with this J first to give rise to what is called the DJ recombination. And then as a second step, one of these V's here, let's just say this one, will recombine with the already rearranged DJ segment, but part right here, this D, and give rise to a completely recombined antibody gene. And this is such that if you recombine this J with this D and this V, you actually get a completely different specificity than if we recombine the same DJ, but say a V here. And so the combinatorial diversity of this cut, paste, and shuffle reaction that is actually ordered is enough to give rise to about 10 to 20 million different antibody specificities. So the germline theory is correct. Actually, I should also point out that part of the way this was verified was to show that in the germline configuration, all these genes, actually, let me say it like this. It's important to note that as you recombine the Ds and the Js and the Vs to the DJs, you actually physically lose bits of DNA from the genome. So what is the intervening sequence between what is recombined together is lost. And so when Tonegawa, who got the Nobel Prize for this discovery, first looked at germline tissue, a skin cell, for example, he found that in a skin cell, this was actually the configuration. But if you were to look at a B cell, he found that in these particular genomic locations, bits of DNA were missing. And that's how the discovery was made. And so that is true then, that antibodies on the surface of B cells are actually recombined. So gene segments exist to encode each antibody once these gene segments are actually assembled. Now this is true, and this could give rise to many, many different specificities, but that doesn't mean that the somatic theory of mutation was wrong. In fact, both are correct, and I'll go through this in a second. Part of the reason that people thought that this was inadequate to explain things is that there exist many organisms for which there are very few germline encoded antibody genes. So maybe 7 V's and a couple of J's, etc. So that would not give you near the diversity you would need at the level of a population of the B cells. What was also known is that even if you could have 20 million different genomically encoded specificities, that 20 million is not uncountable. It's 20 million, and so it would be very hard to suppose then that those 20 million antibodies should be able to take care of every single antigen out there. So it is true then that on top of this recombination process, antibodies mutate even further, but this mutation now is actually induced by the fact that they bind antigen. So antigen goes in and binds for a population of B cells, the one that binds it best, but the binding is improved by the fact that mutation happens in the rearranged variable portions of the antibody genes, the V, D, and J portions. And that mutation then leads to an even larger number of specificities, and the antigen, the microbe, will bind those new receptors and select out the one really that binds it best. And so this was understood genetically, but it was not shown at the molecular level until about 10 years ago now, when Tasuku Honjo in Japan identified a protein, which is a DNA mutator, which is called, he named activation-induced cytidine deaminase, or AID. And what this protein does is it sits on the DNA of antibody genes, and it's a cytidine deaminase, so it will take an amino group away from a C. And when you have a cytidine without an amino group, you really have a uridine. And so by removal of the amino group, a C becomes a U. And now you have not a CG base pair, but a UG mismatch in DNA. And that UG mismatch can be replicated. And when it's replicated, and it's this in the parent cell, and then this, the replication will give rise to two daughter cells. If you replicate from the bottom strand, you generate the original base pairing. So DNA polymerase will go on the top strand, polymerize, see the G, put a C, and go on. But if you polymerize from the deaminated strand, the top strand, you don't generate a CG base pair, but you actually generate a TA base pair, which for this daughter cell, in comparison to the original parent cell, is what is called a transition mutation. And in fact, after Honjo described this, Michael Neuberger in England found out that AID was a very potent mutator, and it would mutate antibody genes, and it would also actually mutate just about everything else. And so he showed, for example, that AID, going back, mutates even bacteria. So this experiment is as follows. These are about 1 billion bacteria that are plated on a plate that contains the antibiotic rifampicin. And by randomly mutating this population of bacteria, have contain individual cells that are resistant to rifampicin. And so you plate 1 billion bacterial cells onto a rifampicin plate, you get a few colonies. But if these bacterial cells contain AID protein, so a vector that has AID that is now induced to express a protein, you mutate better. You mutate about 6 times better. So AID is actually a mutator of bacteria. And if you remove DNA repair systems that will take care of the fact that there's a uridine in DNA, then you mutate even better. And this is quantified here. AID is in a vector that is inducible with a small chemical called IPTG. And so if you plate this bacterial population on rifampicin plates without AID being expressed, you get the same level of mutation pretty much that you get when the bacteria don't have AID in them. But if you induce AID expression by providing the small molecule, then you increase levels of mutation about tenfold. And this is an average tenfold because, as you can see, each dot here represents an individual experiment, and so there's a huge range. So the third Lederberg corollary is that B cells mutate their antibody genes specifically. And we know this to be true now. And we know that B cells do this both by recombining and assembling their antibody genes using a cut and paste recombination reaction. You cut in a D and a J, you glue them together, you kick out a piece of DNA. And or using mutation to then fine-tune their ability to bind antigen. And so here's a population of B cells with antibodies on their surface. antigen comes in. These B cells arose during B cell development in the bone marrow, and the fact that they have so many different antibodies is a testament to the combinatorial power of VDJ recombination, of the assembly of antibody genes. This antigen goes in, binds this B cell X, and stimulates it to proliferate in a reaction that is now known as clonal expansion. And then these B cells, during clonal expansion, will begin to mutate. And as they mutate the receptor specifically, they will generate a family of B cells that is identical because it came from a clonal B cell, but it's different because all these antibodies now are slightly different from one another. They're mutants of one another. And in this situation, then, the antigen X will bind this one better than all the others in the population and select it out. And this last step is called affinity maturation. And I think we still don't really know at the molecular level how this antigen will select out a B cell. So there are many, many open questions left in this field of antibody diversification. A very interesting one, which we still don't really know how to address, is how is specificity achieved? So we now know you have this mutator that can mutate antibody genes, it can mutate bacteria. However, in the human body, it only really is on in B cells and mutates a very tiny stretch of the genome. So how does this happen is still a mystery. Another mystery is how is deamination by AID, the removal of an amino group from the cytidine to make it into a uridine, how does that lead to mutation? There are many, many systems in the human body, at least 4 to 6 that we know of, that are there to take care of the fact that uridine can be present in DNA. Except in the case of the immune system, where in the antibody genes, when you have a uridine in DNA, you always get a mutation. In many other parts of the genome, you have a uridine in DNA, you fix it properly, you don't get a mutation. So another open question is, why are antibody genes so prone to improper repair? And a last and very, very interesting question is, or issue, is the fact that there is a price to antibody diversification. Here you have a huge population of B cells, most of which will mutate. And as I mentioned at the beginning of the talk, mutation is random and therefore it will be detrimental. And in fact, the vast majority of B cells that undergo this mutation process after they interact with antigen will die. 95 to 99% of cells will die. However, it is also the case that almost every single tumor of the immune system, almost all lymphomas, come from errors in this reaction. So if your mutator is not properly tethered to the antibody genes, but goes somewhere else, then that mutation may give you cancers, and in fact it does. So another field that we're trying to address, a lot of us are trying to address, is what is really the price of antibody diversification? Is this mutator going awry in different cells in the body as well as B cells, which also seems to be the case? What is the benefit to a tumor of a mutator going around? Of course, that is a detriment to us, etc. All right, so I'm going to leave it at this for the first part of the talk. This is actually a pretty tiny field of science, but it has entertained a large number of scientists from many different settings. So people who study Drosophila development, bacterial geneticists, immunologists, neuroscientists, in fact, and et cetera, and also bacterial geneticists. And I guess Ehrlich was a polymath and had a hand to play both in antibody diversification and also the second part of the talk, so I'll leave that one alone. So any questions then from you guys on the first part of how mutation is used by the immune system to diversify antibodies so that they can be specifically now able to counter any pathogen they ever see? Yes? So the question is, what triggers recombination in DNA? We do know that recombination is part of a developmental path in B cells, and there is a set of proteins called the RAG recombinase that actually initiate recombination, and that is a very programmed type of thing during B cell development. Interestingly, what the recombinase seems to come from evolutionarily is transposition. So it's the jumping gene type situation that people have described in everything from corn to bacteria. So there is a transposase, a recombinase, that is induced during development specifically.
About platforms, like bacteria, cells will form, B cells will form specific antibodies for bacteria, but do they form them for viruses?
Say that again, I didn't hear that.
Do B cells form specific antibodies for viruses?
Yes, so B cells will form specific antibodies to clear viruses, bacteria, parasites, the works. In fact, one of the problems in virology research, in vaccine research actually, is the fact that people cannot raise high enough antibody responses, good enough neutralizing antibody responses against viruses. And I don't know if you guys heard in the news about the latest AIDS vaccine trial. The problem is people are really trying to raise good antibodies and they can't seem able to, at least not with the methods that people have tried to date. Down there.
Which model organism has most of this been found out?
So mutation is definitely worked on in bacteria, at least first of all. Now, with regard to the immune system, people use mice or human cells. As far as I'm aware, at least in the setting of the immune system, it's mainly those. I'll talk about some other things later. Yes?
Is there like a predictable time frame where you might think that mutation might occur? Like we see that the number of mutants will vary, but then will there be a specific time frame that mutation will start to occur?
So you're asking whether there is a specific time frame for which mutation is empowered to generate this type of new receptors. And the answer is yes. So mutation, not recombination, but actually mutation in the immune system is actually induced by the fact that antigen binds. So once the antigen binds the antibody gene, a set of processes that we still don't completely understand takes place, a cascade of signaling that leads to the induction, the expression of this mutator AID. And so we sort of know how this mutator is induced. It is antigen specific. We don't really know what shuts it off. So some people say that maybe it is a certain number of time measured in a cellular context by population doublings. So there's a certain set of cell divisions after which you turn this off. But the answer is not really known. But during that time from touching the antigen to somehow ending the reaction, that is permissive time for mutation to occur. Yes?
I would assume that the mutator is coded by a certain gene. How much work has been done on finding that gene or set of genes, and what regulates those genes?
So the mutator is in fact encoded by a gene. The gene is called AID. It was found because Honja looked at differentially compared cells that could and cells that couldn't mutate and found the gene. It is not really known what regulates a gene just yet. It also is not known what regulates where the protein goes. And so, of course, you have a mutator, you have to bring it to the antibody genes. It's not known how that occurs. But it is a gene.
And we don't know exactly what allows that mutator to select a certain region.
No, not at all. Any other hands down there?
Cells that are mutated by AID, can they divide and produce memory cells?
Yes. So, in fact, in ways that I certainly don't understand, in order to become a memory cell, most of memory cells are actually mutated. So you have to go through this entire mutation pathway to become a memory cell. So memory cells are usually defined, although they're poorly defined, they're defined as cells that have already undergone AID-mediated mutation. Other hands down there.
Is it possible to change or remutate a mutated gene into, for example, something like process?
Yeah, so it's definitely possible to continue mutating a gene. So the interesting thing that you bring up is that, and you brought up as well, is when does mutation know how to end? Because if you mutate, mutation is random over the antibody gene, so some mutations will screw it up. Most mutations will screw it up, which is why most cells die. But some mutations will make a better fit. And so how do you know to select the better fit before mutation actually goes on and screws that up? And the answer is we don't really know. The idea is that the antigen, that there is a program at which you mutate, you kind of get a little bit outside the area where things mutate to see whether you've actually made now a good receptor. If you have, you don't go back in. You don't mutate again. But if you haven't, you go back in and you mutate, et cetera, et cetera. But this is a lot of hand-waving and also a lot of observational immunology. The molecular basis of this is not known. Yes?
I believe you said that in the HIV vaccine, that it seems that antibody production has not occurred. Well,
yes.
Do you have an idea why that is, that the HIV virus is
Yeah. So nobody knows that. What people do know is that HIV non-progressors, so people who are infected but live with the virus without progressing to AIDS, have very, very good B-cell immunity. They have very, very strong, potent, neutralizing antibodies to HIV, and they don't just have one antibody that they make that can bind HIV. They actually can hit the virus with many good, different antibodies. People are trying to replicate this in vaccines to essentially glean from non-progressors what the reason why they don't progress. The idea is now that there's specific B-cell immunity, and then to replicate that with vaccines. But at least to my knowledge, a vaccine that can really replicate good B-cell immunity to that effect, raise really good neutralizing antibodies, has not been hit upon just yet. I don't think we know why, and people are working on this quite feverishly, as you may imagine.
So it's not that the HIV virus is mutating within the body and therefore eluding the antibodies?
That is partly true, but somehow it is also true that for some reason the antibodies that most of us would make to HIV are not really good ones. And it's not entirely clear why that's the case. So people have, for example, proposed to make to get from these non-progressors the type of antibodies they make, and to then use passive infusion of these antibodies to protect people from HIV. Of course, that's not, really plausible to do on a large scale, but to use that as a therapy. People have proposed that. But what we really need for HIV is a vaccine that will actually raise the right kinds of antibody responses that are raised in these non-progressors. Why is this differential? It's not known. Maybe non-progressors have more hyperactive B-cell immunity, for example. It's just really not clear. Way down there.
For example, the flu, what's is the problem is that we cannot find the specific mutation to actually cure the flu?
Yeah, so that's a very interesting case as well. Neutralizing antibodies can be raised against the flu, but what our bodies usually raise are antibodies against a part of the flu virus that vary a lot. So there are people who raise antibodies somehow against parts of the flu virus that are invariant, and those people don't get the flu, any flu. They don't even need to be vaccinated. But for the rest of us, and we somehow can't do that, most of us, will make antibodies to the flu of 5 years ago. We cannot be reinfected with that flu, but now the flu has changed. So we don't make really good antibodies against all possible strains. But there are people who can do that.
So is it possible to create one for the whole body instead of the whole flu as part of it?
This gets into questions of vaccines that are really not clear to me, in the sense that we usually, so to get rid of bacteria, we have antibiotics, even though we don't really have as many antibiotics as we should or used to, that are actually effective. To get rid of viruses, the only way we know how is to vaccinate, in other words, to hit the organism, the human body, with an attenuated version of the vaccine. The reason we do this is because That's what Jenner did 300 years ago. He vaccinated with vaccinia, and then that was protective against smallpox. We keep doing the same thing, but we cannot necessarily inactivate properly all viruses. So for the flu, for example, for HIV, that is not possible. For flu, every year we see a different flu strain, and so people look at the emergence of the new strain, assume that this is going to populate the entire human race, and then go on to make vaccines. And so there is a lag between identifying the flu strain of the year and the vaccine you make. But wouldn't it be wonderful if people could find other creative ways to raise these kinds? So, for example, I was talking to Dr. McEwen earlier and saying that maybe you should think a little bit outside the box in terms of vaccines as well, and use mechanisms, cells, pathogens that raise a particular kind of an immune response, an antibody response, for example. Maybe we should use those to raise antibody responses to a viral bit, or essentially mainly viruses, as opposed to using viruses to raise an antiviral response. So, but that's a huge area of vaccinology that I'm not really familiar with. Yes?
If you have an antibody that's effective, what prevents the antibody from mutating into something that's not effective?
So an already effective antibody will be selected out of the pool of mutating antibodies, presumably by antigen. This is a little bit hand-waving. And so it will not mutate again. The next time around, that antibody will not mutate at all. It will exist there, and it will neutralize the incoming microbe directly. But there ought to be some mechanism to completely prevent mutation from screwing up an already good receptor. And I don't know much about that mechanism, though it ought to exist. My bet is that it will have to do with signaling. So if you bind something really, really well, if you're already super specific to the incoming pathogen, then the signal is strong enough to the cell that somehow tells it, you're really good, don't bother to mutate. If the binding is not that strong, then maybe the signal somehow induces a cell to undergo mutation. This is not studied very much, as far as I know. There was another, yes?
Is the DNA rather than the mRNA of the cell spliced, does that limit the amount of mutations that are allowed to occur because the DNA genome is permanently being removed?
Yes. So in terms of recombination, the answer is yes. By splicing together different fragments of DNA and getting rid of the intervening sequence, you essentially limit how much more you can do. But that's okay in the case of the immune system, because every cell is clonal. And so the recombination reaction is specific to every cell that comes out of the bone marrow. But it is true, by doing this, you limit the genetic possibilities for that cell coming out, which is why the vast majority of the time, you don't recombine that cell. You need to mutate it. And so by mutating it, you actually first expand it into this huge family, and then you mutate that family of clonal receptors into the one or two or three that actually are better.
So then are there non-specific B cells constantly being produced?
Yes. So there are not only non-specific B cells being made. So the idea is that you will produce B cells that will be able to see an antigen that you will not encounter until you are 95, whatever. So you should be able to see every single thing specifically. The flip side of this is that the B cells are raised in the human body. So what if you were to see specifically your own cartilage? Will you get rheumatoid arthritis? And the answer is yes, you will. And there are safeguards in the immune system before the cells ever leave the bone marrow. They're actually checked to see whether the receptors they made are actually receptors that interact with self. And if they are interacting with self, they're deleted. So the bone marrow and the spleen where mutation takes place is a constant, it's a death field, actually. Most of the cells that rearrange are screwed up and die. And so 95 to 99% of the cells in the bone marrow are destined to die because of this. They're either poorly recombined, they don't make a right receptor, they make a receptor that interacts with self tissue, etc. But those mechanisms are not fail-safe and they do fall apart, and so you have people with arthritis and gout and whatnot. So, yes.
You're studying immunology. What do you do in your lab?
What do we do in lab?
Yeah, for immunology.
So my lab, so I was trained as an immunologist. So in my lab, we do a lot of things that have to do with with trying to figure out how this mutator goes to the right genes, how the mutation it makes is actually not properly fixed, etc. Maybe how to harness the power of this system to make better vaccines. There's also a side, or 50% of the lab does this. The other 50% does what I'm going to talk about. The second part of the talk, which is parasitology.
Do you make up your own questions, or is this something you research about?
Do we make up our own questions? So yes and no, I guess. You can't make questions out of, you can't ask questions in a vacuum. So we stand on the shoulders of many, 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 many people who have made observations in the past, and we do formulate our own questions, sometimes quirky ones, sometimes the same questions that many other people are thinking about, but it's really sort of a, you have to survey the literature, see what's there, see what tickles your fancy actually, and go after that. But of course, you're stepping on the shoulders of many other people to be able to do that. Yes.
Going back to the HIV question of that side of the room, a lot of recent work has been done that shows that people who are resistant to HIV infection tend to have different receptors on their T cells, so that the HIV virus actually doesn't bind in the first place. I would imagine that the better B cell activity would not be the cause, but rather the effect of not having the burden of a high viral load.
Right, so there are multiple ways to be an AIDS non-progressor, HIV infected non-progressor. So one way is, of course, to have a mutant receptor so that you don't ever enter your T cells. But in some ways, in that kind of situation, you just simply never get infected. So your HIV may or may not, since the receptor is very similar, may or may not enter your macrophages, for example. So you don't even know you're infected. You just never get infected. I was talking about a situation where you are infected. Your T cells are loaded. There is viral load in the body, but you actually don't progress because something keeps the virus in check.
I'm referring to a study. Might have been a German study. Someone got a bone marrow transplant from someone who is HIV resistant, and that person seems to right now be, whether or not he's actually free of HIV infection, used to be HIV positive, now he's either free of the infection or very suppressed.
That's interesting.
I'm referring to that. I'm thinking that because the viruses, they do mutate, they could, to a limited degree, bind to that receptor, but it would be very inefficient compared to someone.
So this is true. So if somebody gets a bone marrow transplant, essentially all their immune cells, or certainly their B cells and T cells, are the cells of the donor. So whatever the donor situation was, especially genetically in terms of whether they had or didn't have the receptor, the same situation would be true for the recipient. Yeah.
I also wanted to mention that a lot of the cutting-edge work on finding ways of curing HIV, or at least treating it more effectively, have been done kind of looking past the idea of antibodies in general. Some work has been recently on fusion inhibitors, and there's also been work on mutagens.
Yes.
So we have some built-in, our immune system is not just a mutator, it has built-in mechanisms. There's actually a viral mutator. The only problem is that HIV has something called VIF factor, which can...
So the viral mutator is actually a direct relative of the mutator that mutates antibody genes. We have plenty of therapies for HIV and AIDS. What we don't have is vaccines. So it is entirely possible that if that in terms of therapeutic measures, I think we really have a lot of things that actually work. Inhibitors, I don't know about the mutator, that certainly is part of how cells deal with HIV. To my mind, it also could be something that the virus could co-opt to actually evolve better. So it's possible, because here you have a mutator, it would get rid of 99.9999% of all viruses, except maybe the ones that survive are mutants that are good enough to be better, to help the evolution of the virus, in other words. But there are certainly enough drugs to keep AIDS in check, HIV. Most of those are way too expensive to be used in the third world. And so what I was referring to with regard to HIV and B-cell immunity is the idea of actually making a vaccine, which should be safe and preventative, actually. So you would never even need those cocktail inhibitors, et cetera, et cetera. So they're two, they're related, but some of my colleagues at the Iron Diamond Center downtown would say that, looking for a vaccine is fine, but we have really everything we need to cure this, or to at least make it a chronic disease and keep it under control. So maybe, for example, Gates Foundation money would be better spent in buying these drugs for the third world as opposed to vaccine research. It's a matter of taste, I guess. Yes?
For the vaccines, the HIV always mutates receptors.
Mm-hmm.
What is the correction for vaccines?
So all viruses, all pathogens will mutate. The question is, do you make an antibody to what mutates, or do you make an antibody to what's invariant? And there are bits of the pathogen of HIV, for example, that will have to mutate. But at the same time, there are parts of the viral coat that need to be invariant, because otherwise it cannot hold itself together. So those would be the two kinds of targets, yes.
But assuming there are so many strains of HIV, how is it possible to inoculate a person without actually overloading them?
Yeah. So you wouldn't inoculate with HIV. You would have to find some other way to present to the immune system the peptides of the viral coat that the immune system actually sees. So the immune system shouldn't care if this is HIV or some decoy virus that looks like HIV. The response ought to be the same. But somehow, using decoy viruses, we can't seem to be able to raise good enough antibody responses. So, part of the reason antibodies and vaccines are good is because of the little movie I showed you, that they will go in and through memory clear a pathogen before it actually infects and spreads in tissues. So that's sort of the idea, that it will be great to be able to make a decoy that will mimic enough HIV to raise an immune response, but is obviously not going to infect. Yes?
H1N1, is it a mutation of the common flu?
Is H1N1 the mutation of the common flu? Yes. So H1N1 is a mutation, so flu both mutates and recombines. So there is a shuffling of gene segments, actually, in ways very reminiscent to how antibodies are made. And so, yes, grossly speaking, if mutation is an alteration of the genomic code, H1N1 is a different variant through mutation of normally, what is normally seen as a common flu. Yeah.
How much time it takes to come up with a new strain?
How much time it takes to become a new strain. So these things recombine and mutate all the time in a population. The question really is how much time, how efficient is this recombination for the benefit of a new environment? In other words, you recombine and mutate all the time, but most of the time mutation, because it's random, and recombination will be detrimental. So you would need to mutate your flu to a strain that somehow ought to be able to bind a receptor better, invade the human body better, maybe fly better in aerosols, maybe have better resistance to temperature, which flu is very sensitive to, et cetera, et cetera. So there's a lot of variables that you need to select. But you mutate all the time. But to actually select those variables is also quite random. Okay, sorry.
What happens if a zygote before there is first division undergoes a mutation? What happens?
If something undergoes a mutation very soon after the first division.
I think a zygote, you know.
A zygote.
fertilized egg.
Uh-huh. Well, usually fertilized eggs don't undergo a huge number of mutations. They are, however, recombining through meiosis. And so that gives rise to gene shuffling, in a sense, in different traits. So if you have monozygotic twins, then you do have an almost identical twin, but at the same time, through meiosis, nothing is ever identical. So, yes?
Can vaccination disrupt the natural process of disease development?
Can vaccination induce
Disrupt the natural process of disease development.
the natural complement of, you have to speak up a little louder because somebody just coughed, sorry.
Can vaccination disrupt the natural process of disease development?
Can vaccination disrupt the natural process of the immune environment? Yes and no. No, in the sense that we always raise antibodies against foreign bits and pieces continually. So vaccination is simply yet another decoy pathogen they're going to raise antibody responses to. There is a catch, which is that if that decoy pathogen looks very, very, very similar to a part of a human body, you can breach this tolerance mechanism we talked about a little earlier and get an antibody response now to your joints. So for example, if you have Lyme disease, this is a pathogen that that interacts not only with the antibodies that are produced against it, but the antibodies we produce against it oftentimes interact with joints. There is a chance that you may end up with an autoimmune disease after treatment, for example. So there's always a little bit of a price to pay for the fact that you're raising really good antibodies. On a population level, that's a good enough price to pay. An individual level is a different story. Chances are you'll be fine. You may incur a cost. If you're infected with EBV, for example, you get mono. Chances are you'll never get a lymphoma, but you do incur a rate of getting a non-Hodgkin lymphoma in your lifetime. So do you want to deal with EBV up front? But you have to pay, maybe. So it's always a trade-off. Yes?
You were talking about before price to pay for gene mutation. Were you talking about the harmful effects of natural immunization?
So I was more talking about, so most of the time, as I said, mutation is random, and so most of the time mutation will be actually detrimental to the cell. So if a cell mutates, chances are it will not mutate into something good. If it mutates randomly its genome, chances are it will die. But sometimes mutation, for example, may hit a death gene that will normally induce a cell to undergo apoptosis, and then it disables that death gene. So now mutation is actually advantageous not to the cell, not to the organism as a whole, but to the cell, because the cell will become a tumor. So the price to pay for mutation is, the undervalue mutation is that you do incur a chance of getting tumors. And so the price you pay for having an antibody system that is so effective is that you get tumors of B cells. Okay. Somebody speak up? Down there? Over there? Okay, thank you. Go ahead.
Artificially stop a genetic mutation, for example, like I believe the girl down there was saying how changing the mRNA to limit the mutation. Can you specifically limit that? Like I want to solve from A when A can you limit that mutation artificially?
You ought to be able to. I don't think we have, because all of these proteins that are these mutators, so the mutator for HIV, the mutator for antibody genes, all of these were really discovered in the last decade. So it is not impossible that, and there ought to be, in fact, there are some inhibitors. I'm not aware that have been actually tried in any type of human trials. you ought to be able to limit mutation. All of these mutant proteins are very, very similar with regard to their active site. And so let's just say that you want to stop HIV from mutating, or some virus from mutating, and that virus uses some deaminase molecule like the AID mutator to mutate, and let's say you block that. You may, or you have a tumor that mutates because of AID and you want to block that. The price you may pay is that if your inhibitor is not really tailored to the mutator you want to inhibit, the price you pay is that you may actually harm the immune system. It may not matter if you're old enough and have seen plenty of other pathogens, and you may not need mutation, or you may need to take care of something really quickly before it kills you, and you're willing to live with screwing up this mutation mechanism that gives you a really effective immune system. But yes, in principle, you ought to be able to stop mutation. Okay. Anybody else? All right, so then we'll take a break and we'll come back. Okay, so we'll now, once everybody's seated, we'll continue this conversation with part 2, which a lot of the people who asked questions already have raised, which is we'll talk about how mutation is actually used by pathogens to evade the immune response. Now, we've already talked about the notion of antibody diversification, the fact that B cells are the cells of the adaptive immune system that produce antibodies. The fact that B cells are actually clonal in the sense that each one of them carries only one type of antibody, that they proliferate in response to encounter of a particular microbe or chemical of an antigen, and they mutate in response to the antigen, and then from the pool, the population of mutated antibodies and the cells that carry them, the ones that bind antigen the best are selected and become memory cells and secrete antibody a lot faster the second time that the body's invaded. And so I'm an immunologist by training, and so what I learned through my training is that B cells are unique in their ability to mutate their surface receptors. And I think every biologist you talk to, or many, many biologists you'll talk to, they will always say that their system is unique, and they will almost always be wrong, as I was. So B cells are not unique in their ability to mutate their surface receptors. Many, many organisms, mostly pathogens, also mutate their surface receptors for the specific purpose, actually, of evading the immune response. So we've already touched upon HIV and H1N1, the current flu strain. Bacteria vary their surface receptors to evade the immune response, and I think I mentioned Borrelia. Yeasts do this, and then there are parasites, unicellular parasites like Plasmodium that causes malaria, or the parasite that we'll be talking about in the rest of the talk that causes African trypanosomiasis or sleeping sickness. These parasites specifically change their surface receptors to evade the immune response. And in place of introduction, I will show you a movie of Trypanosoma brucei, how it infects and the types of responses it evades. And as you will see, it specifically evades B cell responses, which is what got me interested in this and what I really am more of a student of at this stage. So this is a short introduction into the parasite Trypanosoma brucei, which is what we study in the other half of my lab. This is a professional code switcher. Here are some parasites swimming amongst red blood cells, and here's a picture of a parasite in yellow and red blood cells in red. These are unicellular, single-cell, flagellated parasites, and they are, I'm not going to say unique, they're special because they are completely exposed to the human immune system. In other words, they don't live like malaria lives, for example, in a red blood cell, or leishmania could live in a muscle cell. They're actually swimming freely in the bloodstream, and as a result, they're exposed to antibodies. The reservoir for trypanosomes is the fly, which is called cheche in the local language, and mammals. So this is mainly wild animals, but also farm animals and, of course, humans. And the means of transmission of this disease was identified to be this fly by Captain David Bruce in the 1890s, and he's shown here. So what happens is the fly bites an individual who's already infected and ingests this quiescent parasite. And in the gut of the fly, this parasite proliferates and develops into the form that will infect humans, which is now resident in the salivary glands. And now, once a fly bites an uninfected individual, like Dwight, it will spit out this parasite, which will then proliferate in the bloodstream, causing disease, and the cycle will start again once a fly bites the person or the animal that's infected, and so on and so forth. And as you saw, infection results in sleeping sickness, which, if not treated, or even sometimes when treated, is fatal in humans, and a disease called nagana in animals, which translates in the local language as loss of spirit. So I got interested in trypanosomes because there has been a long-standing link between this trypanosome, the way the trypanosomes evade the immune response, and parallels with antibodies. So this event of code switching, which we call antigenic variation, was looked at by a number of people in the early 20th century, notably Paul Ehrlich, who I introduced in the first part of the talk as a person with many, many insights into how antibodies are made and what they do. So Ehrlich notes in the 1900s parallels with antibodies, and he actually attempts to cure these trypanosomes. Now, has any one of you worked in the lab, in a tissue culture lab, and used a dye called trypan blue? Okay, so what does this dye do? Okay, so you stain bacteria because this dye just goes into the bacterium, you can see them better.
I was treating microspheres, and so I used to stain alginate and calcium chloride mixture, and so I used the trypan blue to extract the microspheres.
Okay. Has anybody used these to look at mammalian cells in culture? Okay. No? All right. Well, here you can have a trypanosome, stuffed animal. I've used them for the last 20 years to stain mammalian cells. And, there we go. And mammalian cells are stained with trypan blue because trypan blue stains dead cells. Trypan blue also stains bacteria, alive or dead, and it stains trypanosomes. But until I started preparing for this talk to you guys, I hadn't realized that trypan blue is called trypan blue because it's trypanocidal. So Ehrlich developed this dye. It's an organic dye. Turns out it's blue in an attempt to cure humans who are infected, or mice who are infected with trypanosomes. And that's why it's called trypan blue. In 1910, Sir Ronald Ross observes what he called waves of parasitemia in individuals that were infected, and partly for this, partly because of his contributions to the molecular understanding of malaria, or the understanding of malaria, he got the Nobel Prize in 1902. So what Ross observed, and this is drawn again here from a paper of his in the early 20th century, is that if you actually look at the number of parasites in the blood of an infected individual as a function of time, these parasites spike, and there are peaks and valleys of parasitemia. So the number of parasites in the blood is almost undetectable, and then it spikes up, and then it goes down and up. And the periodicity of this was about a week. And what is actually happening, well, actually, I'll go through this a little bit later, but what actually is happening, and I'll show this a little more clearly, is that the number of parasites increases, and then the immune system gets rid of them, and then they switch, and they go up again and again and again. In 1975, George Cross, who's a professor here at Rockefeller, discovers antigenic variation, the molecular mechanism of antigenic variation, to actually be due to code switching. This is a micrograph that George took of the surface of a trypanosome, which is extremely dense and opaque, and it's covered by 11 million copies of a single protein. The single protein he called variable surface glycoprotein, or VSG, and it's switching between genes that carry these variable surface glycoproteins, or VSGs, that give these trypanosomes a new code every time, and that is actually a molecular basis of antigenic variation. So VSG switching, or code switching, is when a host gets infected, it mounts an effective antibody response. It clears most of the trypanosomes that carry the original VSG, but those trypanosomes, as a population, they have a few individuals that switch, and now the immune system cannot get rid of those because to the immune system it's like a second infection. So continuous switching results in chronic infection. This is a graph of this. You start off with a single green trypanosome, you get to a population, you get in that population a cell to switch to red, you get an immune response to the predominant population, and the green cells die because of this strongly neutralizing immune response, but you do now have the surviving red cell. And that red cell will give rise to another population, which genetically really is the same parasite, but it has a different coat. And as a result, the immune system will see the coat as the antigen as something completely different, mount a secondary immune response again and again and again until the individual is exhausted and eventually dies. And so if one were to overlay this on the slide that Ross drew in 1910, what happens is a single parasite from a fly enters the bloodstream, say the human bloodstream, and then that multiplies and spontaneously switches. And so at the peak of parasitemia is when you start having this huge antibody response that gets rid of all the parasites except for the one that has switched. And now that one takes over, gives you another peak, more antibodies are made, et cetera, ad nauseum until the individual dies. And so what I would like to spend the rest of the talk on is some experiments that we've done in the lab in collaboration with the Cross Lab on the molecular mechanism of VSG switching. So each one of the trypanosome chromosomes, each end of the trypanosome chromosomes, the ends are called telomeres, carries this configuration, which is essentially a number of different genes as well as the coat gene. And so there are about 15 to 20, depending on who's counting, of these at the ends of chromosomes. But there are also approximately 1,000 or maybe even more of these in the genome as well. And so what happens is there is a swapping of the original VSG for a new one, and that's the basis for code switching. So the swapping happens in three main mechanisms, which will become relevant later, but I'd like to at least introduce now. One is called a transcriptional switch. That means you have an end of a chromosome here and another end of a chromosome there. As I mentioned, a single VSG is expressed, so there is clonality to these cells. Every cell only expresses one. And that means that out of 1 of 15 ends of telomeres, 1 is expressed and 14 are not. So what happens when you have transcriptional switch is you stop expressing from one and you start expressing from another end of another chromosome. You can also do a thing called telomeric change, which is literally a cut of a bit of the chromosome that contains the VSG and a swapping, a direct exchange for another VSG that resides at another end of a different chromosome. And the last thing you can do, which is actually the predominant mechanism of switching in vivo, is a thing called gene conversion, whereby it is thought that something damages this end of the chromosome. And when something damages a chromosome, you absolutely need a VSG coat to survive. And so it is imminent that it's extremely important that you switch imminently a coat, otherwise you won't make it. And so what happens is, since this copy is damaged, you replace the expressed VSG at the same location with another copy that could be from another telomere or could be from an internal location in the genome. And so this is called gene conversion. So at the molecular level, this type of switching seems very, very similar, or seemed very similar to me with antibody diversification. First of all, there's clonality in the system. There's a single VSG that is expressed in trypanosomes, although there is actually a choice of 15 or maybe more. In antibodies, there is a single antibody gene expressed, and there are at least 2 alleles, and of course multiple configurations of any of those alleles. All VSG expression sites are telomeric, and I didn't mention earlier, but it turns out that all antibody heavy chain genes are also telomeric. So there is something to do with the location. The ends of the chromosomes are special places. There are about 1,000, estimated to be about 1,000 VSG genes. That's about the same antibody gene segments people estimate to exist. expression of the code is absolutely essential for the survival of the parasite. It turns out, though I didn't mention it, that antibody expression is also essential for B cell survival. So if you have a B cell that is now mature, it's out in the bloodstream, and somehow you specifically remove the antibody from its surface, that cell dies. And finally, we know from the field of antibody diversification that mechanisms to diversify are either a cut and paste recombination mechanism that we talked about earlier, or a mutation mechanism. It is not at all clear what initiates switching in trypanosomes, and so we're wondering in the lab whether switching was also a cut and paste mechanism. And so it was already known that switching is independent of the immune response. And it was proposed for quite a while that that switching may be the result of an attempt to correct a damaged bit of DNA containing the VSG. So it was proposed that the switching is a result of a DNA break at both strands, so a double-stranded break at the active expression site. And even though this was proposed, nobody actually tested it for quite a while. And since stupidity is important for scientific research, and so is ignorance, we decided that since we knew we had at hand lots of tools from the immune system, that maybe we should be able to look at parasites using those tools. So what Catherine and Oliver in my lab and George Cross's lab actually asked was, we don't really know. They said, we don't really know what makes a DNA lesion or a DNA break, if that is even the case in a natural infection. But what we're going to do is we're going to make a specific DNA break at approximately the right location. And we're going to ask, does that actually play with the rate of switching? So will an exogenous DNA break increase the rate of switching? And so the experiment, first of all, is to introduce a DNA break into the active expression site. So how do you do this? You have an endonuclease that will sit on a specific DNA sequence and cut both strands of that sequence. And so you also have the recognition sequence of the endonuclease. So the idea was, let's put in this active expression site the recognition sequence of an endonuclease, and then let's express the endonuclease inducibly by adding this small molecule called doxycycline, and then determine that the cutting is at the right place, and then cut and ask, well, what happens to the rate of switching if we specifically cut right at the location of the VSG? And what we wanted to do, what Catherine and Oliver wanted to do, was to determine whether switching is increased, and then also to see how close this artificial switching was to the wild-type situation. How did the cells switch, and how many different coats were produced, et cetera, et cetera. So to actually try to understand whether switching is increased is a problem. And the problem is that really a negative selection problem. So when you start off with your trypanosome, you know what coat it makes, but you have no idea what coat it produces after it switches. It could produce thousands of different coats. So you can't just say, okay, I'm going to make it switch, and then I'm going to see whether it makes X, Y, Z, because you don't really know what it makes, what the switches look like. So the solution that Catherine came up with was to enrich for cells that have switched their VSGs. And this ended up being a problem in the field of parasitology that people didn't really know how to deal with, but it was a problem that was already solved in the field of antibody diversification. And the way immunologists do it is they have a cell with a particular coat. In trypanosomes, the original code we use is called 2-1. And now this large population of 2-1 cells have switched, and they have become, some of them, non-2-1s. We don't know what they are. And then we have an antibody that specifically binds to the original code. And that antibody carries a magnetic bead, and so what happens is you put the original, the culture that is stained with the antibody against the original code, you put it in a magnet. And so what flows through the magnet is some contaminants along with things that simply didn't have the original code, therefore they must have been switched. And then so this is called magnetic activated cell sorting. It enriches for populations that are negative populations. And then Catherine followed up with a technique called flow cytometry, or fluorescence activated FACS sorting. So this is a FACS plot. Has anybody done FACS analysis or know how to explain this plot to everybody else? I'll throw you trypanosome. So the way this works is there is a machine that, from which a very small aperture flows a single cell at a time. And there's a laser that looks at the identity of the cell, and then a magnet that points a cell, if you need to sort it, to different directions in different tubes, depending on the identity of what's on its surface. And so normally you get these two-dimensional plots. On the y-axis here is propydium iodide, which is a death marker, and on the x-axis is a stain of the original trypanosome coat. And so when Catherine put 100 million trypanosome cells through this cone, what she ended up with were cells that were in this quadrant here, which are not positive for the death stain, so they're alive, but they're also not positive for the original coat, and so they're switchers. These cells right here are also alive. They're not positive for propydium iodide, the death stain, but they're also positive now for the original coat. Therefore, they are the contaminants that flew through the system, and there are reasons for that. And so this is a situation with a wild-type trypanosome. This is also a situation with a trypanosome that has the cutting recognition sequence for the cutting endonuclease, but does not have the endonuclease induced because we haven't added the small molecule that will induce expression of this enzyme. And now you induce expression of the enzyme, and all of a sudden you get a huge increase actually in the population of switchers. Again, these guys are live cells because they're not positive for this death dye. They're also not positive for the original coat, and so they're alive and they're switched, and one can enumerate them. And so what Catherine showed is that if you don't have the recognition sequence, but you only induce the enzyme, uninduced and induced, you really don't switch very much. But if you have the recognition sequence and you don't have the enzyme, you also don't switch. But if you have the recognition sequence and the enzyme, your switching increases up to 200-fold. So these switching rates are very similar to the types of rates that you see in a human infection. And so the experiment is, again, to introduce a DNA break in the active expression site and see what happens. And so Catherine already determined that switching is hugely increased. The next hurdle is to decide how switching looks at the molecular level. And that means, number one, to decide what type, to find out what type of VSGs the switching cells are now using and how switching actually happened. And so step one, to identify what is the new code that is expressed at the RNA level, is to do a 2-day experiment called RT-PCR, followed by sequencing, which is essentially to make RNA, reverse transcribe it, amplify it using PCR, and then sequence the product. And this is a 2-day experiment, but in fact, every single one of these techniques, or most of these techniques, ended up yielding a Nobel Prize because at some point they really, really pushed the technical envelope. So what I'd like to do is discuss at least reverse transcription and PCR with you guys, and I'd like a volunteer based on this to explain the RT part of RT-PCR for a trypanosome. Yes, I'd like somebody to explain how RT-PCR works. RT to begin with. Yes.
Normally when you want to transcribe genes, you start with DNA and you make RNA by unzipping the DNA. In this case, reverse transcriptase goes backwards. You have RNA, which is generally single-stranded, and what you do is you put on deoxynucleotides. You attach those to make a DNA strand, and that DNA strand can create a complementary strand. So you just make DNA off of RNA.
Right, perfect. And so here you have the reverse transcriptase enzyme, which is an enzyme that is encoded in retroviruses, actually, and for which David Baltimore got a Nobel Prize in 1975. It sits on an RNA strand. It requires a primer. In most enzymes, a primer is actually a tRNA primer. And then you add deoxynucleotides, and that RNA gets, through the activity of this reverse transcriptase, made into DNA. And at the same time, the RNA, the original RNA, is now deleted. How about you guys pass this on to and so now how about somebody else explains how PCR works? Because that, too, was a technical advance and a Nobel Prize. Anybody for PCR? PCR? Has anybody used PCR in the lab? I am sure many have, yes, down there. Can you explain how PCR works? What's that? Okay, has somebody actually done PCR? Yes. But you gotta be louder because I'm deaf.
General PCR is just used to amplify small quantities of DNA into large workable quantities for actual testing.
Mm-hmm.
You want like a sequence, like an actual procedure.
Mm-hmm.
All right. Well, I guess you would start with your original DNA strand, and you have your primer sequence, which would correspond to whichever polymerase you get, probably stock, if you want to make your own. And then you have your machine for temperature cycling to just promote the creation of new strands from the strand that you started with.
Okay, so there's a crucial bit here, which is you need to do what to the strands before you polymerize?
Wow, the word escapes me, but you need to separate.
Right. So you need to separate the strands. So every time you go through this cycle, you have to separate the strands, and you do that with heat. So you increase the temperature, separate the strands, you polymerize the bottom strand from the X top strand, and the X bottom strand, you make it another top strand, and now you have two new strands, and then you do this again and again and again and again until you get billions of different DNA molecules. And so, because you knew how it was. Well, I have a few freebies too, so let's see. So this is how PCR works, and it's important to know that it's actually, again, something that was pushing the technical envelope so much so that two people got the Nobel Prize for it, actually quite recently. How about sequencing? Can somebody explain how sequencing? What's that? Pick one. So I asked actually this question to a graduate student class, and I said, how does sequencing work? And they said, well, you put the reaction together, and then you ship it to the company, and then you get sequence back. That's how it works today, but that's not how it worked in 1980, or actually even when I was a graduate student. Sequencing? I saw a hand for sequencing. Okay. Yes, sequencing.
The DNA gets broken up into various sizes, and the smaller sizes of DNA, small sequences, get pulled by electrophoresis faster than the larger. And then it would be looked at and broken up again, and then attach the phosphorus to it, and to see what base each one ends in.
Okay, so it's a little bit backwards. You start polymerizing the DNA, and then what happens? Because you need to, what happens after you polymerize? I'll tell you how it used to be when I was doing this. It's now done very similar ways, but not quite. Anybody else wants to? This is good, but it's not exactly right. Anybody else? Sequencing? So what happens is you polymerize. In the past, you used to have 4 tubes. And each tube has all the DNTPs, because you need the building blocks of DNA. And then each tube also has a dideoxynucleotide. So DNA polymerase cannot attach anything. There is nothing to attach a new base to dideoxynucleotides. And so what happens is you have the tube with a DDA, and the tube with a DDT, and the DDG, and DDC. So 4 tubes for the same template. And so because there was more of the normal DNTP than the dideoxy, you had a whole bunch of different populations coming out of each one of those reactions. And each one of those populations would extend up until a random point where you insert the dideoxy. And so you would then separate, like you said, on a gel, the different fragments of DNA, each one of which, depending on one of your 4 reactions, would have stopped at an A, at a T, at a C, or a G. So then you would look from the bottom of the gel up, and you would say, I'm going to read the sequence now. This is an A. Then the next one up is a C. And the next one up is a G. And the next one up is a T. And you go on across. This is very similar to how the companies do it today, except the dideoxys are actually fluorescent. And so when you get a little track of peaks, those peaks are colored. And so your A's are usually red, and your T's are green, and your G's are black, and your C's are blue, etc. So you can read it on a single track. All right, so the point I wanted to make here is that this is a 2-day experiment nowadays. But that experiment is something that we stand on the shoulders of all these people that pushed the envelope and generated these techniques. So Nobel Prizes are given for techniques as well as for conceptual advances, although this, one can argue, is in fact a conceptual advance as well. So in this 2-day reaction, what Catherine and Laura found is that, going back to trypanosomes that switch, that the DNA break we made in these trypanosomes has led not only to switching, but to switching that was extremely diverse. So each one of these had the full complement of DNA, but like I said, they're clonal, so they only expressed one kind of RNA, and by sequencing that RNA, what they found was that they got many, many different kinds. So this is good, because that means not only that the DNA break increases the rate of switching, but in fact it leads to diverse switching, which is what happens in vivo. And then what Oliver wanted to know is what happened to the original VSG. And so to do that, so actually let me back off. So these are the types of things that can happen to the original VSG. There is transcriptional switch, as I mentioned earlier, where you turn off transcription from one end and you turn on transcription from another end of another chromosome. There's telomere change, there's gene conversion. All of these have different outcomes at the DNA level. So in all of these cases, there is a new RNA, a new coat, a new protein being made from a new RNA. But in this case, at the DNA level, your old coat gene is retained at the same location, but you've actually turned off transcription of that and turned on transcription of another one. In the case of telomere change, obviously, a new VSG is made, the old DNA for the old VSG is retained, but it's now switched locations. So for example, instead of, let's just say, the end of telomere 1, it has now swapped into the end of telomere 3. And the vast majority, but so I should say here, if you think about this, you have 15 possible locations, let's just say, so that gives you a chance for 15 possible switching events. That's not enough coats for the trypanosome. If you do this, you can swap these ends for one another, and by doing that, you get, let's just say, 15 times 15, which is still not enough. So the vast majority of gene conversion, of switching, happens through a mechanism called gene conversion. Now that is interesting because what happens is somehow the original copy is damaged, and as I mentioned earlier, if you don't have a VSG, you die. And so there is pressure in the system to pick up a new coat and put it in the original location. This happens by copying from an intact copy into the original location. And so as a result, even though there is a new RNA that is expressed, the new VSG, the DNA for the old VSG, this guy, is lost. And that can be copied from another telomere, or it can be copied from an endogenous location. But the main point is that the old DNA is gone. So Oliver decided to look at this based on these 3 assumptions on whether your DNA is there, whether it changed places, or whether it's not there at all. And he looked at this by a technique called rotating agarose gel electrophoresis coupled to Southern analysis. So this RAGE technique is essentially like running a DNA gel, but a very long one, so that you separate intact chromosomes. And I'll show you this in a second. What happens then, which was again a technical advance in the early 70s, is something called Southern analysis, which is named after the discoverer of this, whose name is Ed Southern. And what Southern decided to do was to transfer, to put a bit of paper, nylon membrane, on the agarose gel and let it sit there for a while so that the chromosomes, by charge, actually transfer to the nylon membrane, or the DNA transfers to the membrane. And then you can do a lot of things with a piece of paper that you can't do with an agarose gel. For example, you can probe the membrane. It's called probing. What we really talk about when we say probing is we take a bit of DNA that is identical to the code protein, actually complementary to the gene of the code protein, to the gene of the code protein. And then we label that bit radioactively, and then we essentially dump it on the membrane. And what's going to happen is that bit of radioactive DNA is going to go and find its partner on the membrane and light it up. And we can do this, as I'll show you, for the initial VSG, and we can do it for the new VSG as well. So this is what this looks like. This is a RAGE gel stained with ethidium bromide. These are the many different chromosomes present in trypanosomes. There are the megabase chromosomes, what's called intermediate and mini chromosomes. And now this can be transferred on a membrane. And the way Oliver transferred these was grouped by the VSG, the RNA that is now expressed. So we start off with a code called 221, and this group of switchers now express a code called 224. And so this is at the RNA level. At the DNA level, however, here you have your parental line with DNA for 221, and that DNA is completely absent from all the clones. At the same time, if you probe the same membrane with DNA, a bit of DNA that will probe for the new code, you find that that is present in the parental line on a single copy here. And it's also present in the switchers, but in a double copy. And in fact, this new copy, if one lines up the two blots, lines up exactly with where the original DNA was. So not only have we lost the original DNA now that we make a new VSG, but we've actually duplicated the new VSG into the location of the original gene. Yes? These are actually clone numbers. It doesn't really mean anything. Just this is clone 3, 16, 18, 19, etc. These are as you pick them out of the plate. Really, the only point to remember is that, and this is a technique that people have used in trypanosome research for a long time. And I'd be very interested in knowing what happens to the antibody logs, for example. Nobody has looked at this in antibodies. So the only thing to note is that the copy of the original DNA is gone, and the new RNA that is being made is actually, the DNA for that RNA is actually duplicated in the same original location of the old DNA. So that means that a DNA break doesn't just increase the rate of switching, an exogenous DNA break, but it leads to switching that is high in diversity, and also switching that leads to a loss of DNA, and this can only be what people call duplicative gene conversion. So VSG switching after artificial breakage is then very similar to natural switching in terms of repair processes utilized. And so there are many, many open questions left in this field. This is a field that will really benefit from a lot of investigation. For example, one of the questions we're dealing with now is, so we have shown that if you make a specific DNA break, you can mimic the real situation. The question, that doesn't mean, however, that that in a wild-type infection, the wild-type infection switching is actually initiated by DNA break. It could mean that. So we're very interested in trying to understand whether there exists DNA breaks in natural infections and what the mechanism that makes those actually is. And then another thing we're very interested in is, I showed you how these original 2-to-1 code switches to 2-to-4, switches to many others. It turns out that the switching in trypanosomes is non-random. It's very reminiscent of VD-J recombination in antibodies, where DJ recombination happens first and V2DJ recombination happens next. When you start with the known code and you inject this in a mammalian host, what you get out at the first peak of infection is quite predictable. And then the second peak of infection is also quite predictable. So there is a hierarchy that somehow serves this organism that we don't understand what allows for it mechanistically. All right. So in the second part then, we talked about how mutation is used by a specific pathogen called T. brucei to evade the immune response. T. brucei is a unicellular parasite it is special because it completely is exposed to the humoral immune response to antibodies. Now, we generate phenomenal antibody responses to this parasite, but then the parasite switches its coat. And so our phenomenal antibody responses are useless. The general mechanism of switching seems quite similar, broadly speaking, to the mechanism that antibodies use to diversify. Natural switching can be recapitulated in vitro by introducing a DNA break, but we don't know what would generate such a break in vivo, though we are actively looking into this. And then in the first part, we talked about the parallel in the immune system, how mutation is actually beneficial in the immune system in order to generate B cells and their antibodies, how B cells, like trypanosomes, are clonal, how they proliferate in response to encounter, how antibody genes mutate in response to this binding, and how pathogens like the Brucei or many other viruses and bacteria would select out of this pool the B cells that bind them best. And many of these become memory cells. So here you got it. There is, in fact, directly a battle of the mutants. This is a micrograph that is false stained of parasites amongst red blood cells along with white blood cells. This could be a B cell, for example, that saw the parasite and is now blasting, is becoming huge in order to generate antibodies. And so these cells are going to generate antibodies to the parasite, and the parasite is going to switch, and then the antibodies are going to be for naught. So that's pretty much what I wanted to talk about today. But before I finish, a science talk is never complete if thanks and credits are not given to many people. Specifically in my case, I would like to thank George Cross. I know a lot of immunology, but I am still very much a student when it comes to parasitology, and so George is my teacher. He's probably one of the most prominent parasitologists still active today, and most likely, if you encounter a parasitologist in your travels, there's a very high chance that that person trained with George. Also like to thank many of my mentors and teachers. I'm not going to mention names, except to say that you guys are here, many of you with your teachers. Take a moment to thank them, because if you ever end up in science or in medicine, it's going to be because they sparked your interest in this. So thank your teachers. All right. I'd like to thank people in my lab, especially, and the Cross Lab, especially Catherine and Oliver, mainly because, so Oliver taught us a lot of things that we really had no idea about, and Catherine was brave enough to pick up a completely different topic that really my lab was not working on until the last 5 years or so. I'd like to thank everybody at Public Affairs at Rockefeller, especially Gloria, Zach, and Tanya, and the whole team of people that took time off their holiday break to be here and set this up for you guys. And I would be amiss to not thank Michael Duschenko, who actually took this lovely picture and allowed us to use it for this presentation, as well as YouTube for the 3 videos. Okay. Oops. No, too far. Okay, so now to questions. Yes? Say again? Guys, you gotta be quiet because I can't hear.
When the fly infects the
Mm-hmm.
is there like a specific nutrient in the blood that allows it to produce?
You know, I have no idea. And I've been wondering, it's too bad that George couldn't make it here because maybe he knows the answer to that. But being an ignorant student of parasitology, I haven't actually read anything that explains how the switch happens from the parasite in the midgut of the fly, which is actually not switching, in fact, to what happens to the parasite in the bloodstream, which begins to switch. And so I've been wondering about this. I've been wondering whether, and I don't know if there is an answer to this or people just haven't looked, because it's actually quite a small community. So I've been wondering whether In the blood, there is something that the parasite likes that makes it switch. Or I've also been wondering whether it's as simple as a temperature change. So the fly is usually 25, 27 degrees. Human body is 37. So maybe it's as simple as a temperature change that induces this kind of switch. I don't know the answer to it. I don't know if anybody knows, but it's a fascinating topic to look into. Yes?
Does the fly suffer from the antibody disease or not?
No, flies are fine. I don't know if flies are in any way damaged by the trypanosome. Yes, down there.
And VSGs change their original form?
VSGs?
Change their original form.
Do VSGs change in the original form? What do you mean? Do you ever go back to the original VSG?
Sorry.
I'm not sure I understand.
Like when they first enter the body and then change, can they do that again?
So not if the change is by duplicative gene conversion, because then you actually lose that DNA from the genome of the trypanosome. But if it's by another mechanism, yes, you can change back. It would not be advantageous to the organism, the population in the human body, because we would already have made antibodies against that coat. And these antibodies are super good. So if it switches to that coat, it's dead. Yes?
You said like multiple coats are showing at the same time in the bloodstream. Could the immune system mount like antibody responses to multiple coats at the same time?
Yeah, so the immune system can mount immune responses to multiple coats, but usually there is a selection. So this is a very balanced system, very finely tuned. So there are not that many new coats at any given time. There's also selection by the immune system. So at every peak, you probably see 3 or 4 or 5 different coats for which the antibody, the immune system makes antibodies. But that's not enough. Then they switch to something else. Let's get somebody from here.
So that when the antibodies move from the surface to the cell, the cell dies. What is that?
When a B cell doesn't have antibodies on its surface, it dies. This is absolutely true. I don't know why that happens. The idea out there is that the receptor, the antibody, allows for a baseline level of signaling to the B cell to say, hey, I'm a B cell. So the antibody is actually the reason of being of the B cell. And if you lose the antibody, you actually get confused and eventually you die. So people call this tonic signal. That there's a level of signaling from the receptor that leads to the survival of the cell. But that's very descriptive. Yes?
You drew a parallel between the mechanisms that create variation in the B cell with the mechanisms that create variation in the trypanosome. Is there a mutator gene in the trypanosome like you described with KIV? And if that's the case, has anyone ever created a sort of a knockout culture of trypanosomes that can't change their code?
Right. So the answer to that is that's actually what got us into this in the first place, that maybe there's a mutator that does this for the trypanosomes. We actually don't know what initiates this switching in trypanosomes. It would be phenomenal if there were a mutator gene, and we would lovely to find it. Presumably, if that becomes the case, yes, then you can knock it out and you have no switching. But the hypothesis we're currently working on in the lab is that maybe you actually don't need anything to initiate this except for the fact that the VSG, the code genes, happen to be at the telomere. And telomeres break all the time. So we're thinking that maybe it's this natural breakage that is random but frequent enough to be a stimulus for this. But if there were a mutator, that would be great. Yes? What causes telomeres to break? There are many reasons telomeres break. First of all, so telomeres are very special. They're at the ends of the chromosomes, and they are actually very hard to replicate. And there is a specific protein called telomerase that is charged with extending and replicating telomeres. And in addition to this, that they're actually fairly fragile and very well protected, at least in mammalian cells. In addition to this, here you have a situation where you have to actively transcribe through the telomere because that's where your VSG is. So now you have potentially, this is all hand waving, you have the transcription machinery going through. You have some type of DNA replication trying to go through as well. You have a collision. And although this is completely hand-waving, there are many reasons why. So I don't know which is the right reason for trypanosomes, but there are many reasons why ends of chromosomes are very fragile. In fact, one of our faculty here, Titia De Lange, just recently shown that telomeres or ends, DNA right close to the telomeres can be considered a fragile site, which is a very cancer-prone site also in the human genome. So I'm not sure why they're so fragile, but they are. Yes?
So you know how the trypanosome, am I saying it right? Okay. So you know how they change coats and everything? Is there a point when they just run out of combinations?
Yeah. So that's a good question. So as an immunologist, I was always taught, as I told you guys in the beginning, that the immune system can make antibodies toward anything. And so there's no way for a pathogen to stump the B-cell response. And here you have a trypanosome that exhausts the B-cell response. So I don't know if, in terms of the African trypanosomiasis disease, I don't know whether you eventually die because you've exhausted the immune response, because trypanosomes can make a huge number of coats. Or rather, my inclination would be that maybe the number of coats that trypanosomes can make is limited, but maybe they switch so fast that the immune system needs to make a response every 7 days, which it cannot handle. Because all of these cells, if you remember from the beginning of the adaptive immune response, are actually quite slow in growing. So they require a certain amount of development time. And if you hit them, every 7 days, they may not make it.
So in essence, like if you know like all the combinations, for example, and if you create like some sort of a vaccine, for example, that's probably not going to happen. But so then you could prevent it afterwards, like if once you know it. Yeah.
No, I understand what you're saying. So I would imagine that there are billions of different combinations, very similar to the numbers of different combinations of antibodies presumably one gets. But I don't think that anybody has actually mathematically modeled how many possible combinations there are, partly because the genome of these critters was not completed until, I believe, 3 or 4 years ago. So this is, really new stuff. Somebody from up here. Yes?
Say, for instance, someone's infected, right? What steps do scientists and doctors take to, get this virus that changes codes to save the patient?
Say it again, sorry.
What exact steps does doctors and scientists take to save a patient who's been infected with a virus that's codes change?
A virus or a trypanosome or anything. So what steps do you take to cure somebody who is under, is diseased with one of these things? A lot of the drugs that were developed for trypanosomes are extremely toxic. They were developed 50 years ago, 30, 40 years ago, and in many cases, once actually the parasite goes out of the bloodstream and crosses the blood-brain barrier, there is really not much you can do. That is to say, the drugs that you take to cure it have about a 10% mortality rate. So 10% of people will die just by taking the drugs. So there is really not much you can do. If you catch it before it crosses a blood-brain barrier, there are some drugs that are also very toxic, but they could eventually clear. So this is actually a real danger if you guys are, going to leave tomorrow to go to sub-Saharan Africa on a safari. You should worry about flies. Yes?
I have a two-part question. The first part of the question just goes to, you said that the genes that code the certain proteins are by the telomere. So, as you say, telomeres break off. And I'm also assuming that these surface proteins have another function besides just getting past the immune system. I'm surprised that if there were, I'm assuming that there is another function, important proteins would be coded at the telomere.
So antibodies are telomeric as well. So telomeres break off all the time, but as long as you have copies somewhere else, you're actually okay. I don't know, I guess I can't ask a trypanosome, so I don't really know what other function these VHGs serve for the trypanosome. I would actually say that maybe they actually served some other function a long time ago, but right now they just, at least as far as we're concerned, they're a coat for the immune system. A trypanosome needs a code. It doesn't need a VSG code when it's in the fly. So there's no switching when it's in the fly. So that makes me think that the actual switching of the code is something specific to its niche, which is a bloodstream.
And the second part of the question is that first of all, could we say that the fact that telomeres do break off and that the trypanosomes do have to bring in other coded proteins to kind of replace it, as we've seen, would that be considered a main facilitator of that switching? And if so, if we use telomerase on trypanosomes, could we get in the way of their switching?
Yeah, so trypanosomes have active telomerase all the time. And in fact, one of the people in the lab has knocked out telomerase, or in collaboration with the cross lab, and we're trying to find out whether if you don't have telomerase, and therefore you can't really do much, you cannot extend your telomeres. So your telomeres get really, really short, and your breakage now becomes a little more important because it doesn't just break the telomere, but it breaks a little bit further into the genome and damages the VSG, whether that is going to be enough in a tissue culture situation to increase switching. But again, what we do in the lab doesn't necessarily have to be the case of an in vivo infection. But yes, we're looking exactly at that. Yes?
Two questions. The clip you showed us in the beginning of the lecture, how do you think the man survived from
I don't know, actually. I would imagine that he took one of those very toxic drugs that helped him clear the parasite. So if you remember, trypan blue, which is an organic dye, was used successfully by Ehrlich to treat a couple of people. Probably killed another few, but you know.
How does exercise relate to the immune system? How does it support the immune system?
What supports the immune system? Oh, well, I don't know. Does somebody down there want to answer this question? I'm really not a physiologist or a neuroscientist at all. In fact, it's too bad that you're asking this question now, because Dr. McEwen studies exactly all these things, how stress influences the immune system. There's an interplay of physiology, the nervous system, and the immune system. There's crosstalk. All the studies that show that exercise, for example, strengthens the immune system are correlative studies in populations. I don't know. I'm sure there's going to be an underlying mechanism, that is to say, well, when you exercise, you release endorphins. Endorphins go back to this and that, the other thing. But I don't know that. If anybody has actually looked directly at that, I'm sure people have, actually. Yes? Yes? I'm under the impression that it's actually he's taking drug inhibitors, cocktails of drug inhibitors. So you can. Well, one of your colleagues up there can explain better than I. Pedro, would you like to explain how drug inhibitors keep HIV in check? Would you like to explain to your colleague down here how drugs against HIV actually keep AIDS in check?
Well, it depends on which antiretroviral we're talking about. There's some that are called fusion inhibitors, and what they do is they get in the way of the virus being able to bind to receptors on your cell. Say you have a T cell, it has receptors. Say you have a CD4 cell, CD4 cell has CD4 receptors, and what HIV does is it will bind to them. The fusion inhibitors prevent that from happening. There's another kind of inhibitor called reverse transcriptase, and that prevents, HIV is an RNA virus, it's not a DNA virus. In order to infect you, it has to turn its RNA into DNA and implant DNA in your DNA. In order to do that, it uses reverse transcriptase. What a reverse transcriptase inhibitor does gets in the way of that. There's some that get in the way of the enzyme reverse transcriptase. There are others that actually sort of make nucleotides into the gene as it's producing DNA. It stops the whole process of producing DNA. The only problem is, of course, that HIV constantly mutates. So it gets, you can get past those.
Okay. There you go. Yes. All right. Sure. Good. Okay, you get a screen.
Every time I have seen representations of receptors or antigens and its interactions, they've always been represented with models, as of course are atomic reactions. And I was curious if with the newer advances in electron microscopy, tunneling microscopy, etc., if people have actually ever visualized these interconnections, or if things are still being done at the level of models.
People have actually visualized the connections with fluorescently labeled antibodies on the surface of cells, and then you cannot see them with microscopy like this as individual full-fledged receptors. You can see them like this as crystallography models, etc. But you can actually visualize at the surface of cells the receptors. You put an antigen or another cell type, and you can see them. It's called capping. They all stream from the surface of the cell to that one location to bind the particular entity. So you can see that in fluorescence microscopy, time-lapse microscopy now.
What about with viruses, when viruses actually enter into another cell? Is that also actually, has this also actually been observed in some direct?
Yes, and so that has been observed, again, very, very recently with different types of fluorescent microscopy. So you can see, for example, a little red virus kind of bumping along a little phylopodium, and then eventually it reaches the body of the cell and fuses in. But all of those are very recent visualization techniques, but yes.
I'm amazed at how much is in advance since I was in school.
Tell me about it.
I asked you if your lab was willing to take on one of the high school students.
So I have plenty of high school students in the lab, especially in the summer. We're a little bit limited in physical space right now, but yeah, we're always open. So, all right. Maybe the rest of the questionnaires can come down here. Thanks, guys.